Howdy folks, Mr. Ulrich here. I'm just going to take a couple minutes to review the parts of the cell that are important for us to know if we're going to talk about cell division mitosis. By the end of this video, you should have in your notes a diagram of both a dividing animal cell and a non-dividing animal cell and have the different parts labeled. I'll also take a couple minutes to talk about the structure of a double chromosome. All right, get yourself a nice blank sheet of paper. Doesn't have to be unlined. It can be lined, doesn't matter, notebook, but give yourself a full sheet. We're going to do two cells on this and label the parts on the inside. The first cell we're going to do is a non-dividing cell. This is the cell that you're most used to seeing. All right, and this is, we're going to do an animal cell first. I'll do a plant cell later on. And right away, you can, of course, see the reason why I tend to do most of my notes in PowerPoint because my handwriting isn't so good. But anyway, the non-dividing cell is a cell, like I said, that you're probably most used to seeing. So draw yourself a nice fried egg. Woo! Fried egg. And give us a nice big healthy yolk here in the middle. All right. And when you look through a microscope at a cheek cell or something like that, or an amoeba, this is what you're going to see, right? Now, I often will put little dots in here. That's part of the cell called the cytoplasm. Not because you're going to be able to see individual structure. Sometimes students ask me, are those ribosomes? No, you wouldn't be able to see ribosomes. I'm just showing that there's a viscosity. There's a difference between inside and outside. And that difference is going to be maintained by the cell membrane. That's really going to be the job of the cell membrane. All right, less to protect the cell, which is what a lot of my students come to me understanding. And though it's not necessarily wrong, really its main job, like I said, is to keep the inside different from the outside. And underneath that is, like I said before, the cytoplasm, literally cell stuff. Right. In your textbook, they'll describe the cytoplasm as the watery matrix of the cell that all of the organelles are floating in, right? That really doesn't give it the credit that it should have. Really, this is where most of the metabolic reactions of the cell are going to be taking place. So all of the chemical reactions of the, within the cell are happening essentially in the cytoplasm. So all of the raw materials, all of the products, all of the enzymes for all of the intermediates of all of those chemical reactions. So it's pretty complex. It has no structure. It's like stew. Anyway, we'll just write site of metabolism. All right, but let's get into the nucleus here, since this is really the storage place for DNA. A lot of students will come to me thinking that, describing the job of the nucleus as the control center of the cell, like there's like a person in there driving the cell around. No, really all the nucleus is the site of DNA storage. Right, cytoplasm, not a super friendly place for nucleic acids. They're going to break down out here over time, and so us eukaryotes keep it nice and safe, all our DNA safe inside of the nucleus, right, within the nuclear envelope. And I'll just put that down here. Nuclear membrane, nuclear envelope, right? This is the boundary of the nucleus. Right, we're going to find that in, like I said, eukaryotes, but that's later on. Now, let's take a look at the nucleus itself. All right, before we just fill it in, right, color it yellow like it was a fried egg. Now, let's think of this nucleus here as a dinner plate. And for dinner, we're having spaghetti meatballs. So draw some spaghetti. Spaghetti. And put a couple meatballs in there. All right, you can put 1, 2 veggie meatballs if they need to be, whatever, 3, doesn't really matter. All right, these, well, inside here, the spaghetti is the DNA. It's in what's called chromatin form. Chromatin. All right, so chromatin is loose and stringy DNA. More importantly, it is readable. Loose, stringy, readable DNA. In fact, the only time DNA can be transcribed into RNA, the only time that replication can take place is when it's in chromatin form. And then inside the meatballs themselves, right, are the nucleoli. All right, nucleolus for one. Nucleolus. I describe it as the little old nucleus, right? So you take the word nucleus, right, and you just put a little "ol" in the middle, right, of the nucleus, and you get the nucleolus. This is where RNA synthesis, right, RNA nucleotides are going to be synthesized there. And those are the parts. Oh, I forgot important structure that we're going to find in animal cells. They look like little exclamation points. You wouldn't be able to see these in a light microscope. These are way too small, too weak, can't see them. All right, but I'm drawing them bigger for our dining and dancing pleasure. All right, these are the centrioles. All right, and this cell has two of them because it's going to go into cell division. It's about to go into mitosis. So let's take a look at the structures that we're going to see. What the job of the centrioles. Sorry. The job of the centrioles is to organize another structure that's going to become super important here as we get into cell division. Structures called spindle fibers. So speaking of spindle fibers, we need to draw our dividing cell because that's where we're going to see those spindle fibers. So in the dividing cell, things are a little bit different. So dividing cell, and remember it's an animal cell. I didn't leave myself quite enough space to draw this. All right, but whatever. So let's give ourselves at least the outline of another fried egg. All right. Because things are going to be a little bit different in this dividing cell. For one thing, in order for the cell to divide, all of this DNA has to be replicated. And then all of that DNA also has to be organized so that we can make sure that we get one set of DNA in each resulting cell. Right, so the structures that we're going to find, we're not going to find loose stringy chromatin because that's like crazy, right? Organizing spaghetti when it's all cooked and loose and stringy. So instead, we take all this information and pack it up into little notebooks. Right? You can almost think of your A and B day notebooks. A lot of students will keep two notebooks, so they'll have all their A day classes in one notebook and the B day classes on the other day notebook. Then it's really easy to bring one or the other notebook to school and not have to bring both of them. But now we're talking about, instead of 4 classes worth of notebook stuff, we're talking about 3 billion base pairs that need to be organized and not make any mistakes. All right, so instead of dividing individual genes or the spaghetti here, they're going to be organized into chromosomes. And these are, I'm going to draw the X chromosomes that you know so well. So just draw a couple big X chromosomes like this, right? Give me another one. They can be kind of haphazard, right? They don't have to be oriented any particular way. I'm going to draw a small pair of X chromosomes, and here's another one. All right, give yourself an even number. All right, and that'll become important later on. All right, so here are the chromosomes. You'll notice there is no nuclear membrane. Right, you'll notice there is no nuclear membrane. The nuclear membrane is gone. All right, and all that chromatin has been condensed down into these chromosomes. Now, another thing that's taken place is the centrioles here are going to move and migrate. All right, they're going to go towards either pole of the cell. Right, remember the cell is round; it's not two-dimensional. Right, and so we'll often use the same references that we use on a globe, right? The poles and an equator. That'll become important when we talk a little bit more about mitosis, but let's get through these parts first. In between these structures, you'll see, and you can see these right on the in a microscope. Right, you won't be able to see the centrioles. Again, I made those big so you can see them. But these are the spindle fibers, okay, which are protein filaments that stretch in between the centrioles. So let's label some parts here. All right, of course, we know the cell membrane and the cytoplasm. I'm not even going to label them here because we got them labeled up here. There's a couple other things that we need to label. So let's label the chromosomes. Right, and this is a double chromosome. Excuse me, and that's going to become real important later on as we talk about mitosis. So this is a double chromosome. This is condensed DNA. Right. The centrioles here have moved. And you can see they would. I learned these as asters. These they look like flowers. But we don't need to know that. What we do need to recognize is that these are called spindle fibers. Right, the centrioles are organizing these spindle fibers. Now, the spindle fibers are going to provide a mechanism to move the chromosomes. Each chromosome is going to be attached to a spindle fiber. And it's going to be the interaction between the chromosome and the spindle fiber that's going to allow the chromosome to move, the genetic material to move to either side of the cell as it divides. So the spindle fiber is going to allow for chromosome movement. Right, no nuclear membrane, no nucleus, right? The centrioles have separated and the spindle fibers have organized. Another structure that I should take a quick moment here to look at a little bit closer is that of the double chromosome. All right, the double chromosome is really, and I'm doing it with a zigzag here on purpose, the way I'm drawing it here. All right, because really what each leg of the doubled chromosome is, the chromatid, right? Remember, this is the double chromosome. This right here is a chromatid, right? The single leg of the chromosome. And in the middle, right, where they're attached, is called the centromere. All right, the point of attachment between two. And this is important. This is where the spindle fiber is going to interact with the chromosome here. All right, and it's also important for us to recognize that each chromatid, right, is a single long strand of DNA. It is the result of replication, so these are equivalents. They have the same thing, and like I said, you can grab onto one end and unravel that if you wanted to. Well, I'll stop it there. If you have any questions, please make sure that you drop me a line either through email or put it right in Google Classroom. Other than that, I'll see you guys in class.